Welcome to this introduction to quantum biology. Today, we begin a journey into one of the most exciting frontiers of modern science, where the strange laws of the subatomic world meet the complexity of living systems. For decades, biology was seen as a field of statistical laws and classical mechanics, but new research suggests that life doesn't just tolerate quantum effects, it thrives because of them. Understanding this is key to unlocking the secrets of enzymes, photosynthesis, and even how birds navigate. Hello, I'm Diego, and I will be your guide through this amazing field. In this first lesson, we'll establish what quantum biology actually is, and why it's a departure from the classical biology you might know. We'll look at the pioneering ideas of physicists like Erwin Schrödinger and see how their order from order principles still resonates today. Our goals today are threefold. First, to define what we mean by non-trivial quantum effects. Second, to explore the established mechanics in enzymes and photosynthesis. And third, to look at the cutting-edge research on how quantum processes might actually drive biological evolution. Quantum biology is the study of specific phenomena like tunneling, coherence, and entanglement within living cells. We're not talking about the basic quantization that governs all matter. Every chemical bond is quantum mechanical, and that is alone trivial. We're looking for cases where a quantum effect does functional work a classical description cannot explain. And we're asking how nature maintains such delicate states in the wet, noisy, warm environment of a cell, which is close to the worst possible place to try. These ideas aren't new. Pioneers like Niels Bohr and Pascual Jordan were asking if atomic physics was essential for life as early as the 1920s. They suspected that the indeterminism of the quantum world might be amplified within living organisms to influence the entire system. It was Erwin Schrödinger's 1944 book, What is Life?, that truly set the stage. He argued that classical order from disorder laws, like those governing gas in a balloon, couldn't explain the high fidelity of genetic inheritance. Genes are simply too small to be governed by classical statistics. Schrödinger proposed that life relies on an order from order principle, where incredibly small groups of atoms are safeguarded by quantum theory. He envisioned the gene as an aperiodic crystal, that is, a complex molecule where every atom plays an individual, vital role in the organism's instructions. One of the most established proofs of this is in enzyme catalysis. Enzymes accelerate reactions by enormous factors. Most of that acceleration is classical. The enzyme stabilizes the transition state and lowers the activation energy. But that isn't the whole story. Enzymes also facilitate a process called quantum tunneling, where particles like electrons and protons pass through energy barriers rather than surmounting them. Lighter particles like hydrogen are particularly adept at this quantum shortcut, and hydrogen tunneling in enzyme reactions at physiological temperatures is now well established. What remains debated is how much of the catalytic power tunneling actually explains, and whether enzymes evolve features that promote it. That argument is live, not settled. Now, moving from enzymes to plants, we come to photosynthesis. the most famous claim in this field, and the one where the story has changed the most. When light hits a leaf, it creates an exciton that must find its way to the reaction center. For about a decade, the story was that the exciton samples many paths at once in a superposition and instantly finds the most efficient route. Nature, it seemed, had built a room-temperature quantum computer inside a leaf. That specific story did not survive, and the correction is more interesting than the original claim. The experiments were done on the Fenna-Matthews-Olson complex, a pigment protein structure in green sulfur bacteria that acts as a molecular wire. They reveal long-lived oscillations, and those were read as electronic coherence. But the superpositions of exciton states, right there, survive no more than about 100 femtoseconds, while the oscillations persisted beyond 600 femtoseconds, two numbers that far apart cannot describe the same object. In 2017, a landmark paper put the conclusion in its title: Nature does not rely on long-lived electronic quantum coherence for photosynthetic energy transfer. Here is what replaces it. The long-lived oscillations are real, but they are vibronic, hybrid states where the electronic excitation and the protein's own nuclear vibrations mix into one inseparable superposition. The protein isn't noise that the coherence survives in spite of. Its vibrations are part of the quantum state, screening the excitation from the environment and slowing the coherence. Photosynthesis doesn't fight its warm, wet surroundings, it recruits them. And the efficiency that actually needs explaining isn't speed, it's directionality. Nearly every exciton reaches the reaction center instead of escaping or recombining. We even see these effects at a microscopic scale in bird migration. The radical pair mechanism suggests that birds detect the Earth's magnetic field through quantum entanglement. Light-initiated reactions in the bird's eye create entangled states sensitive to the magnetic field's angle. You'll often hear this described as entanglement. An entanglement is present in the models, but the functional quantity is a spin correlation. What makes this case compelling is that the theory predicted something strange before anyone could test it. Weak radio frequency fields in the megahertz range should scramble the compass, while other frequencies should not. And it was borne out. Noise between roughly 0.1 and 10 MHz disrupts avian magnetic orientation, while noise up to 100 kHz does not. Cryptochrome-dependent magnetic senses have since been demonstrated in fruit flies, cockroaches, and plants. So, the radical pair mechanism is a widely accepted account of the avian magnetic sense. Widely accepted is not the same as closed, though showing that this specific radical pair is the compass a bird actually flies on is a chain still being completed. This brings us to a cutting-edge area of research and to my own corner of the field, quantum evolution, where researchers suggest that quantum mechanisms are not just for metabolic processes. They might be fundamental to how life explores the vast landscape of genetic possibilities. Most of the attention in quantum biology has gone to metabolism. Far less has gone to evolution itself. In this framework, genes are viewed as digital objects. Because a gene is a discrete sequence of nucleotides, a mutation is not a smooth slide but a jump between discrete states. The proposal is that the DNA molecule could exist in a superposition of mutational options. with the cell acting as a measurement device that monitors its own code. To test the search question, this research uses n-dimensional genotype networks and compares classical random walks with quantum walks, and here the result is clear. Quantum walks are significantly faster and more efficient at finding the adaptive solution, regardless of how many mutational steps the search begins from it. That's what you should expect, because quantum walks spread ballistically rather than diffusively. But I want to draw a careful line, because two very different claims are easy to blur. The first claim is mathematical, and it is not speculative. On these networks, quantum walks outperform classical random walks at search. That is a robust result. The second claim is biological, and it is speculative. Whether evolution actually does anything like this inside a living cell is a separate question, and our simulations do not settle it. It would require the superposition to survive long enough in a real genome in a real thermal environment to matter before the coherence closes the window. That assumption is not established. Quantum evolution remains a promising but speculative research program, and I say that as someone who works in it. The suggestion is that evolution, it seems, has optimized its own exploration tools. There's also an appealing conjecture here, and I will flag it clearly as a conjecture. The efficiency of quantum walks might help explain why the principle of maximum parsimony works so well in phylogenetics. If evolution followed the most efficient path, it would naturally leave behind the simplest mutational histories we observe in nature. That's an idea. It is not yet a tested one. We also see potential quantum effects in olfaction, and this is the most contested case in the field. The vibration theory of smell, in its strong form, proposes that our receptors detect the vibrational signatures of odorant molecules through inelastic electron tunneling, rather than simply recognizing their shape. If that were true, a molecule and its deuterated twin should smell different, because they vibrate at different frequencies while keeping nearly the same shape. That was tested directly in 2015, and receptors did not distinguish them. The paper's title was blunt: Implausibility of the Vibrational Theory of Olfaction. What survives is a hybrid, sometimes called the striped card model. An odorant needs the right shape to bind the receptor, and then the right vibrational frequency to trigger the signal. Recent theoretical work is consistent with that picture, but its authors say the honest thing plainly. The recognition mechanism remains primarily unresolved. So olfaction is not a success story of quantum biology, and it is not closed case either. It is a live and genuinely unsettled problem, which I think is a more interesting thing to teach. How do we test these theories? One powerful method is isotopic substitution. By changing the mass of atoms, like replacing hydrogen with deuterium, we shift both the mass of the tunneling particle and the vibrational frequencies, and then we watch what happens to the rates. Tunneling depends steeply on mass. Classical barrier crossing does not, in the same way. So the kinetic isotope effect, and especially the way it depends on temperature, becomes a fingerprint. Looking forward, the lessons we learn from quantum biology suggest incredible new tech, from high-efficiency artificial light harvesting to quantum simulators modeled on biological systems. We are beginning to understand the wet and noisy coherence that nature has been working with for 3.5 billion years. To recap, quantum biology reveals that life operates at the intersection of classical and quantum laws. We have seen how tunneling contributes to enzyme catalysis, how vibronic coherence and not long-lived electronic coherence underlies energy transfer in photosynthesis, how a spin correlation in radical pairs may give birds a magnetic compass, and how quantum walks explore networks of genetic possibility faster than classical ones. Whatever that turns out to mean for real biology. We have briefly covered the historical foundations, established biological mechanisms, and the latest research on quantum evolution. This field reminds us that life is far more interconnected with the fundamental laws of physics than we once thought. I hope this lesson has deepened your appreciation for the quantum nature of life. As you reflect on these concepts, consider which of these cases you find most convincing and what evidence would change your mind. Also, consider how these quantum mechanisms impinge on the nature and fundamental ways in which biology operates. Thank you for joining me in this introduction to quantum biology. I will see you in the next episode on olfaction and sensory perception. Always remember to be curious as children and stay Renaissance.